of pros and cons, of rods and cones, and spectral sensitivity. Part number next, on photoreceptors. So there are rods and cones. You read about that, or at least you heard about that, right? What's the difference? What's the difference between rods and cones? What are their roles? Like, do they do the same thing in the eye or slightly different things? And where are they sitting in the retina? Like, is the retina full of both at all places of it, or are there some parts of the retina that are more like that have more cones and some parts of the retina that have more rods? Try to think about these three questions, pause the video, think about it, then compare your answers to mine. Let's look at the answers. Rods and cones have different shapes. Rods are shaped like rods, and cones are shaped like cones. That's a little bit of an exaggeration because they both have cell bodies and a large part of both cells are kind of similar, but then at the end, in one case you have a longer thingy that has the same width along it, and you call it a rod. And then some other type of cells has a shorter thingy that also gets more narrow as you go away from the cell body, and these are cones. Do they have different concentration in different parts of the retina? In our eyes, yes. Here's the actual arrangement. In the fovea, we mostly have cones. In the fovea itself, we don't have any rods at all. And then close to the fovea, we mostly have cones, but we start to get a little bit of rods as well. But the further you go towards the periphery, the more rods you get. Here's a nice summary. It's a little bit of a busy plot, but let's figure it out. The x-axis here is the distance across the retina measured in degrees, because it's round. So the part that you're looking at, the direction of your gaze, is the 0 point. This is where you're looking at. And then stuff that's 10 degrees away. So 2 degrees is the size of your thumb if you look at the length of your arm. 10 degrees is like it's the width of your hand taken at the length of your arm, something like that. So 10 degrees away from the fovea is like this part. Like this would be 90 degrees from your fovea actually, right? 90 degrees away would be perpendicular to the direction of your gaze. It would be like this part, and that's almost the part where our visual perception ends, because if you think of the light, it's impossible for it to get like over here. Probably about 90 degrees, like here, is the furthest it can go, the most peripheral vision we can have. Okay, so this is distance across the retina, and we go through the point where our fovea sits. Good. Then this axis is actually the number of cells, the prevalence, and we have rods shown in red and cones shown in blue. In the fovea, we only have cones. Rods drop to 0. Can you notice that? And that's pretty weird. That's pretty narrow and wild. Fovea is only cones. Then as we go away from the fovea, the number of cones gradually drops down in both directions, here and here, while the number of rods increases, at least at first. From no rods, we go to a lot of rods nearby. As we go even further, the number of rods also decreases because in general our peripheral vision is very bad, so it just goes down, and there are almost no cones there. And on top of that, we also have the blind spot, which is like literally just a gap in both of those. No cones, no rods, just a blind spot. This part, nerves and vessels entering and leaving the eye, remember? But this has two interesting consequences. One, the fovea is only cones, and cones, what is the difference in function? Do you remember? What do cones do? What do rods do? Okay, so which ones do we use to see in color? Cones. Cones allow us to see in color, and rods are kind of black and white. So do we have one type of cones or several? And do we have one type of rods or several? If cones can see in color, and there are different colors, it kind of implies that there are several types of cones. We'll talk about it in the next slide, so let's park it for now. And all our rods are of the same type. It doesn't have to be like that. Fishes, for example, and in frogs, they have two different types of rods, but we have only one type. Okay, good. Cones give us color vision, and in the fovea we have only cones. See, it's written here and shown here. Cones are shown in green, and cones in the fovea are particularly tiny, teeny tiny, tightly packed because we need high resolution vision over there, right? Why do we have rods then? If cones give us color vision, isn't color vision kind of cool and amazing? Why would we use rods? Do rods have a strength about them? Yes, they are more sensitive. I'm not sure how to relate the shape of these two cells, like the conical shape of cones and the rod-like shape of rods. I'm not sure how to relate this shape to the function of those. I suspect that there is a way, but rods are longer and they are more sensitive to light. So essentially we have two systems. One that requires lots of light but allows us to see in color, and another one that can work with very little light, but it's black and white, and also it's noisy. That's another thing. Because it's tried to detect even the tiniest amount of light, it is kind of noisy. And also because the fovea is picked with cones, it means that our high resolution vision is in color, or the other way around, that color vision has high resolution. But our low light vision, like the vision that you use if you're walking in your backyard at dusk, this vision has low resolution. And even more interesting than that, because there are no rods at all in the fovea, it means that when we're staring at something, we can only perceive it with cones, which means that in the dark, we're blind exactly in the middle of our visual field. And then here we have a little bit of a division, I guess, between people who actually walk in the dark and people who actually don't. But if you do it, then you're acutely aware of that. The best way to see something in the dark is look not exactly at it, but near it. In the middle of your visual field, you have a blind spot. Essentially, when you're walking in the dark, you have two blind spots. Our normal blind spot, but also the fovea, usually the best part of our vision, becomes a blind spot. One way to notice that is to look at the sky, at the stars, when the night is clear, and if you live in a place or happen to travel in a place with low light pollution, then you can go, and if you can see the Milky Way, then you should be able to also see some fun stuff like star clusters and even some nebulae, maybe. But the thing is, like with an Orion nebula, or this most bright star cluster called the Pleiades, you actually see them better if you don't look at them. Like stars are bright enough for us to see them with our fovea, and we can see their color, right? From the fact that Jupiter is yellow and Mars is kind of orange, you can figure out that we can see the color of stars. There are red stars like Antares, and then there are blue stars and white stars like Sirius. But for example, the Orion nebula that's sitting around here, or even the Pleiades, which is a star cluster, like these are the Pleiades in this case, you see them better if you look not at them, but nearby. So you would be staring approximately at this point, and then you will see this thing with your almost peripheral, like almost central but still a little bit peripheral vision. Then you will see the best. Try it at some point. Go out and try to find them in the sky and see how your vision changes in the dark. But so let's talk about cones now. Cones, we use them for color vision, which kind of implies that there are several types of cones, right? Because in order to detect different colors, we need to have different types of cones. How many different types of cones do we have? We have three: short wave, medium wave length, and long wavelength. Light is waves. They're much faster than sound waves, and these are not oscillations of air or anything, but somehow they are waves in some physics, quantum mechanics sort of way. So we can characterize different photons or different kinds of light with its wavelength. Nobody talks about frequencies of these waves, by the way, because they're too high. But wavelength is actually important. We won't talk about the actual values, it's too specialized for us, but let's just at least remember that shorter wavelengths have high energy and are, for example, bluish, and longer wavelengths are kind of more relaxed and are kind of reddish. You can see that over here. Shorter wavelength blue, longer wavelength red. If it's even longer, it becomes infrared. We cannot see it. Snakes can. if it becomes even shorter, it becomes ultraviolet. Again, we don't see ultraviolet, but bees and birds do. But let's return to the cones. We have 3 types of cones: short, medium, and long wavelength. How many primary colors do we have then? What is a primary color? Primary colors are those colors that you can mix and create new colors, but you cannot create a primary color from other colors. How many different primary colors do we have overall? 3. And there seems to be some logic here. 3 cones, 3 primary colors. So let's talk. What are these primary colors? Blue is one, red is another, and the next one is either green or yellow, depending on the context. We'll talk about it in a second. So let's first discuss this situation. What cones are activated when we perceive red? The long wavelength cone, right? Because look, these are the longest wavelengths. This is the sensitivity curve for the long wavelength cone. It's not particularly sensitive to short wavelengths, but it is sensitive to longer wavelengths. And if you look at the red colors over here, it is basically the only cone that has sensitivity in this range. Does it make sense? So if we have something like this red coming to our retina, the short wavelength cone will be silent, the medium wavelength cone will be silent, but the long wavelength cone will be responding to it. Okay, so red is L cone. Good. What about blue? Same but opposite, right? If we have blue color, M cone is mostly silent, L cone is mostly silent, S cone is responding. So we have some logic. L means red, S means blue. Now, how does green look like? M cone is sensitive to green, mostly. So if we have green coming, S cone is silent, M cone is active. What about L cone? It turns out L cone is also active, but not as much as M, right? M cone is more sensitive to green than L cone. All right, so if we have S, it's blue, L is red, M, mostly M, is green. What about yellow? Yellow is weird. For yellow, we have both L cone activated and M cone activated. So if we have a lot of M and a little bit of L, that's green. If we have a little bit of M and a lot of L, that's yellow. From this picture, from the picture of how responsible the cones are to light of different wavelengths, we can figure out how they get activated. And because we know how these wavelengths are perceived, we can build sort of a table of correspondence of how the brain has to process this information to achieve the sensations that we have. Because these wavelengths we perceive as yellow, we know that. We can just show this wavelength to a person and ask them, what color is it? And they will say, yellow. But also we know that it's a lot of L cone activation and some M cone activation. We can figure out that that's the language that the brain has to be speaking. When L cones are active a lot, they don't spike, but they talk to the neurons that eventually spike. So if L cones are active and M cones are also active but less, it will be perceived as yellow. Which, by the way, means that if we mix some green and some red, we can also activate M cones and L cones in such a proportion that it will be perceived as yellow, even though we won't have the wavelength that correspond to yellow. We'll have a mix of blue and a red. And that's how color mixing works. And there is a link in the description to a video called This Is Not Yellow. If you haven't watched it before, watch it. And if you have watched it before, still watch it, because it's good and relevant to this part. So let's reiterate that. We infer colors from activation of different types of cones. And because there is an overlap between these curves, colors that are in between activate more than one type of cone. Like a yellow color activates both green cones and red cones. But it means that mixing some green and some red will give you a perception of yellow. And it also means that mixing some green and some red and some blue in appropriate proportions so that all the cones would be activated equally would give you the perception of white. What is the white color? White is no color. White means the bright light of the sun, not in the evening when the sun is yellowish or reddish, but kind of in the middle of the day. The sunlight is our default lighting. That's the default color that everything has. A little bit of the blue sky, a little bit of the yellowish color of the sun mixed together give us the perception of white. White is the default for our vision, and that's a mix of green and red and blue. And if we introduce these primary colors and mix them in an appropriate proportion, we will have a perception of white color. Now, if you really recreate white from basic colors in a theater, you will have these funny colored rainbowy shadows, but that's a different story. And that's basically how all color mixing works. In the theater, when you're lighting a scene, and also in our screens, all colors are synthesized from green, blue, and red, according to three colors that activate our cones the best. If you take a magnification glass and bring it really close to your screen, you will see these individual glowing pixels. The more modern the screen is, the more fancy it is, the tinier the pixels. In a phone, for example, screens are really high resolution, so pixels are really tiny, but still, if you have a droplet of fluid on your phone and this droplet acts as a tiny lens, you can totally see the pixels, or you can see them if you just bring it close to your eye and then really carefully like stare at it. When you print things, everything is slightly different. In print, you use magenta, yellow, and cyan, because in this case you have what's called a subtractive mixing of colors. With lights or in the theater or with pixels on the screen, you add colors directly, like you have this type of light and then you add another type of light on top of it. When you make a book and you use dyes, each dye reflects certain wavelengths but absorbs, like eliminates, squishes, silences other wavelengths. So if you mix two, then you are left with a color that is neither of both. It sounds a little bit weird, but if you think about it, you will understand. Yellow, for example, yellow dye means that it reflects green and red, but totally cancels blues. While the magenta means that it reflects blues and reds, but totally cancels greens. So if you mix magenta and yellow, then the only color that both of them reflect is red. And that's why mixing magenta and yellow in a typographic process gives you a red. And that's how books are printed. But we won't dwell too much on it. If you spend some time on this topic, you can kind of reconstruct the logic of it. And I totally advise you to do so and intuit the differences between additive and subtractive mixing, because it helps you to understand colors better. It's a good thing to invest to, broadens your horizons. But we'll talk a little bit more about biology. So let's look at this plot of sensitivities of different rods once again. Let me ask you the following question. You know, there are mutations, right? Every now and then we have a mutation in one of the genes, and sometimes the gene is broken, sometimes it's just slightly altered. And obviously, we have mutations in those genes that make different cones function, in those genes that make a cone a blue cone or a red cone, in those opsin molecules that actually catch the light and respond to it. If you were to guess, which of color separations is most vulnerable to mutations? Like, which colors would we be most likely to lose due to mutations? If you know that the sensitivities of cones follows this chart, which colors seem to be most vulnerable? Pause the video and think about it. What do you think? Which of these separations is most vulnerable? Blue looks pretty different from either red or green, right? It stands kind of aside, but the green cone, the so-called green cone, the M cone, and the so-called red cone, the L cone, have lots of overlap. Look at that. Green is just M more than L, while yellow is kind of L more than M. So we have two different colors, but the distinctions are tiny. This all feels so subtle. This must be the vulnerable one. And indeed it is. The most common color blindness is red-green color blindness. Shifting any of these curves, either the M cone or the L cone, just a little bit closer to the other one, makes them too close for the brain to separate them. So the most common color blindness is called deuteranomaly or deuteranopia. And it is really common. 5% of all males have it. And here I use males in terms of genetic males. 5% is a lot, which means that 2.5% of the entire population has it. Then there are other types that together sum up to about 3%. So overall, color anomalies, color blindness are pretty common in the population, and we should be cognizant of that. And by the way, what can we tell from the fact that genetic males are more susceptible, much more susceptible to color blindness? Genetically female humans with color blindness are very rare. What does it mean? It means that it has something to do with X and Y chromosomes, right? Because, in genetic females, you would have XX chromosomes, while in genetic males, you have XY. And if genetic males are more susceptible to this, you can suspect that maybe this difference is involved, and indeed the problem is that this gene sits in the X chromosome, of which genetic males have only one copy. So if this copy is faulty, there is no backup, while for genetic females it's a little bit easier because of 2 copies of an X chromosome. And this is a test for color blindness. Depending on whether your vision is normal or abnormal alternative, you will see different numbers here. I believe there are 3 options. You either see 57, which is normal vision, or you see, I think, 35 if you have one type of color blindness, and maybe you can also see nothing at all if you have another type of color blindness. And if you carefully investigate how this test is built, you can kind of figure out the logic of it. To a person with a normal vision, some of these little bubbles will look like bubbles of different colors, while with a person with abnormal vision, this difference will disappear, but some other difference will actually become more distinct and more noticeable, and this would lead to difference in overall perception. And the biggest consequence in practice for a person who writes reports and analyzes data is that, remember, the red-green color scale, color map is not a good one. So if you ever prepare a report and you have a map or some color charts, the green to red gradient is not a good one. To a person with normal color vision, it's almost upsetting because the difference between the green and the red is not only obvious, it also feels like it has an inherent meaning to it, right? Like green is like the leaves and red is like the blood. So it's life and death. It sounds like a good metaphor, but 5% of the population won't be able to see this map on the left. Just they won't have any idea about which part is greener and which part is oranger, because both would project to them to some sort of generic brown. And a fix for that is very easy. One option, of course, is just to use red and blue. But another option is to add a little bit of blue to either green or red. So you just don't want to have a warm red and a warm blue present in your map. You either do like green and cold red that has a little bit of blue mixed in, or you have warm red without blue and kind of teal emerald shade of green. That would also work, because then the blue color will remain even if the green-red difference is lost. And these days you can look online and there are so many nice pre-packaged, prepared color maps, and for many of those there is almost like a rating of how colorblind friendly they are. If you ever find yourself in need of a good combination of colors, one good place to start is colorbrewer2.org. I really encourage you to go to, even just for inspiration, just to figure out how good colors look like for data visualization and data presentation. So an interesting question, if you're not colorblind, or if you are and you're wondering how bad is it in terms of like art and stuff, because colors are important, right? So are there colorblind famous painters, for example? Are there any famous painters that would use color a lot but at the same time would be colorblind? What do you think? Yes, the answer is yes. So which really famous painter was probably colorblind? We're actually not sure, but it looks like that was the case. That really depends on how well you can imagine different art pieces, but let's think. Is there any famous painter who would paint in color, but their colors would be kind of curious? Then let's see, if they are red-green colorblind, it means that they would use red and green kind of interchangeably, and they would use blue and maybe violets for color contrast. Do you know anyone like that? Van Gogh, of course. We aren't actually sure that he was colorblind. It's not quite clear, but it looks like he probably was. Another problem with his paintings is that he used pigments that were new in the era, and many of them turned to be not quite stable. So many of his paintings probably look very differently now than they looked when he painted them. But the differences are still consistent with the color blindness theory. Like some of the things that are bluish now used to be purple, for example. Some of the things that are orangish now used to be more red. But it looks like he used red and green pretty interchangeably, while blue and purple were a very different color for him. So, it's definitely not a barrier for being a genius painter. But so let's talk about that a little bit. M and L cones are vulnerable to mutations. Why are they so close then? Like, why? Is it good? Is it a sort of payback for some optimization? A side effect? A sad consequence that we have to bear and tolerate because something else is amazing? Is it the optimal placement of sensitivities? What do you think? Is this good? Like, is it a good way, this plot over there? Is it a good way to place sensitivity curves of different cones? Again, step into the shoes of Archangel Gabriel, if Archangel Gabriel wears shoes. If you were to redesign cones from scratch, how would you have placed sensitivity curves? Think about that. No, the answer is no. That is stupid. This is a stupid distribution of sensitivity curves. Here how a good distribution of sensitivity curves looks like, the lower one, in a European starling, which is a bird. You want them, whatever number of curves you have, to be distributed at equal distances from each other. Then you will have the best possible color separation. And look, this bird also has an ultraviolet cone. So here's already a hint. Birds are much better than humans as far as color vision goes. Birds can see more colors because instead of 3 cones, they have 4 cones, but we'll return back to that. Look at the humans though. Again, in the human, blue cone is separate. Good. Green and red are too close to each other. Oh, by the way, look at that. I think they have a typo in this paper because the green and the red cone colors are flipped. Ha ha ha, we found a mistake in some paper online because I just copied it from a paper online. But okay, what can we do? Anyways, the green and red cones in a human are too close. Placing them at equal distances would have been a much better choice. This is horrible. But then the question is, why? Why our cones are so badly placed? How come normal species, like lucky species, have their cone sensitivities, opsin sensitivities, placed optimally along the spectrum, while in us, blue is okay, but green and red are just sitting together in this really vulnerable and uncomfortable way, so that instead of just relying on simple rules like this cone to this color, this cone to this color, we have to look at the relative contribution of M and L cones and from that make judgments about whether it's red or orange or yellow or green. How come? What does that mean? Why our cones are so suboptimal? So look, this is a tough question. It actually requires some thinking. You can figure it out from your common sense, but you have to be brave and bold and also use everything that you know about evolution that we talked about before, and also take into account the fact that an optimal distribution is actually possible. If we look at the birds, we know that it's possible. So one option that this is somehow impossible is out immediately, right? It is totally possible. It's just that for some reason our cones are crappy. Well, not the cones themselves, but the opsins, the way the opsins are separated. What can it mean? Find an explanation for that. How come we have this really suboptimal distribution of cone sensitivities? Pause the video, think about that, put an effort in that, come up with a hypothesis, and then let's compare your hypothesis with the actual story, which is kind of interesting. Here's the story. Originally, vertebrates had good color vision. Birds still have good color vision. In birds, there are L cones for red, and then M cones for green, S cones for blue, and then special ultraviolet cones for ultraviolet. And if we look at reptiles and salamanders and all these animals that stemmed from some proto-vertebrates, they all tend to have 4 cones. However, mammals typically have not just 3, but 2 cones. Only 2. The middle and the short wavelength. Mice have 2, but also sheep and deer and cats and dogs and horses, all of them only have 2 types of cones. Sometimes you can read about how they're colorblind. So they're colorblind relative to a human in the sense that they don't perceive 3 primary colors. They are like most colorblind people who have 2 primary colors. But they still can see colors, just fewer of those. And basically, they cannot differentiate between green and red. But primates, including humans, somehow have 3 types of cones again. Just 2 of them are really lousy. They're too close to each other. And if you think about this evidence, you can come up with a story. And actually, if you look at the molecular structure, at the genes that encode these opsins, these special proteins that actually catch light, you get a confirmation for this story. It's very similar to this story that we had with taste receptors and also smell receptors. Remember how humans had lots of smell receptors, but most of them were pseudogenes, as opposed to, say, dogs that had many receptors, many smell receptors, but most of them were functional. If you don't use a certain gene, babies don't suffer when they are born with a mutation with this gene, and so genes can become pseudogenes, ruined, dysfunctional versions of a gene that are no longer relevant, and then at some point they may be eliminated. And ancestral mammals had lost not just 1 but 2 different cones. What does it mean? What can we tell about its life? So in which case you don't care about colors? What style of life do you have to have in order not to care about colors? Nightlife, right? If you live at night, or if you live in holes, which is pretty similar, but if you live in low light conditions, then you don't care about vision that much. What will be your favorite types of sensation? What senses will be most important to you? What senses are most important for a mouse, for example? Hearing, touch, and smell. So see, it kind of fits the story of smell as well. These ancestors of ours used smell a lot and didn't rely on vision that much, at least not on color vision. They were not mice. Mice are modern animals. Mice are not among our ancestors, but our original mammals were probably kind of mice-like in their look and their ecology. If you think of shrews, have you seen a shrew? Look it up at some point. Shrews are not mice at all, they're not rodents, but they have kind of vaguely similar shape. They're tiny and they make holes and they catch worms and little bugs, and that's what they eat. And they're mostly active at dusk and at dawn, maybe a little bit at night, or maybe during the day if they don't have to leave the hole. So probably that's what our ancestors were. And the big, pretty creatures that walked the earth had good color vision, and they were not us. But then something happened. Do you know what happened? Have you heard of anything that would kill all beautiful, pretty creatures? Anything that would have happened in the past that would allow tiny cold dwellers survive, but kill all the beautiful daytime creatures, especially those large ones? Yeah, right. It's called Great Extinction, the meteor that killed the dinosaurs. Not all dinosaurs died. Some dinosaurs didn't. The smaller ones, have you heard of them? They're called birds. Birds are essentially dinosaurs. But big dinosaurs died. When the meteor crashed into the Earth, and it was huge, maybe it was a comet, it hit somewhere near modern South America, and half of the world burned, and then there was no sunlight and no summer for a few years. So lots of things died. There were no summers, there were no plants, there was no food for big animals. Only small animals survived. Our ancestors, who hid in the holes, just had enough rotting stuff lying around to survive these few years. And tiny dinosaurs that eventually became birds also survived because they could fly, I guess, and they could like find some food. But all the big beautiful creatures were gone. And then once they were gone and the life kind of normalized, more or less, those tiny little ancestors of ours were pretty much the only one left, them and birds. They started to differentiate, and some of them became larger and some of them became more powerful. Some of them became predators and started eating others, but all of them went through this period in their life history when they lived underground and didn't care about light. And so now we have this strange situation when most large predators that walk the earth, like lions and tigers, have only two cones, and most grazers that eat grass, like sheep and horses, have only two cones. We lost our color vision. And then primates reinvented it from scratch. And from this transition from a typical mammal to a typical primate, actually typical old world primate, you can see how it probably happened. The middle cone mutated, and from one type of cone we got two subtypes. We had a slightly more long wavelength cone and slightly less long wavelength cone. So if your answer was something like it's still evolving, it's not perfect because it's not perfect yet, this is basically a correct answer. They are so close because they re-evolved from scratch. Not quite from scratch, but almost. We only recently, in the evolutionary history, reacquired trichromatic vision. We're still not at the level of birds that are tetrachromats, that have 4 different types of cones, but at least we have 3. And again, when I say we, I mean humanity in general, even though some people are functionally dichromatic. And the story of how it evolved exactly is complicated. It's written up there. It looks like at first they mutated and then it duplicated. I always somehow assumed that there was a duplication of genes, so we got 2 versions of the gene and then one of them became slightly different. It looks like it's actually the other way around. First, 2 varieties of gene emerged, and then in old world primates it duplicated. In new world primates, it's still not duplicated, and so only females, and not even all females, but most females are dichromatic, and that's because of this X chromosome linked position of the opsin gene. That's bizarre. Like imagine in new world monkeys, not all monkeys have color vision, but only most females, because monkeys are smart enough and they can figure out like that Julie can, see in color and we can ask Julie if necessary. That's weird. But yeah, evolution is interesting. And why did it evolve? What's the point? What's the benefit? Why didn't it evolve in sheep or in tigers or in dogs, but suddenly primates, specifically primates, reacquired trichromatism? What do you think? What's the benefit of being able to distinguish between blue and red? Berries and fruits. Also maybe predators. But let's start with berries and fruits. Why berries exist? If you're a plant, why would you have a tasty baby? Because fruits are plant babies, right? A seed inside is the new plant. So why would you, as a plant, develop a tasty baby and then place it somewhere and make it colorful? What's the point? Yeah, because if you're a plant, you can't walk and your baby cannot walk either. So if you make your baby tasty, but it's not actually the baby, it's more like the crib. So your baby is hard and made of wood, essentially, but the crib is really tasty. And you put it out there, and who will come? A bird will come. A bird will come, and the bird will eat the berry, and then the tasty crib will be, used by the bird because it's sweet and awesome. But the kernel, the little seed, the bird will just poop it out, right? The bird doesn't have teeth. Most birds don't. Birds don't chew generally on what they eat. So if they swallow a baby, if they swallow a berry, they swallow it whole and then they poop out the seed. And indeed, if you ever try to get rid of poison ivy, for example, you would know that it just keeps regrowing back, because birds like poison ivy. They eat it. It's not poisonous to them. They eat the berries and then they sit on the tree and they poop out the seeds. So every year poison ivy reappears under every tree where it can grow, because that's how birds carry seeds around. Birds are good, but for most fruits, mammals actually aren't good, aren't helpful, because we have teeth, and with our teeth we can damage plant babies. And so plants just evolved into this beautiful way of hiding their fruit from us, from mammals. They made them red on the green background, because you know what? We cannot tell. I mean, sheep and bears actually cannot tell whether red is red. That's kind of wild if you think of how the world looks like to most animals. Like a tiger, say a picture of a tiger over there. A tiger is orange, and you may think, why would someone be orange if they are surrounded by greens? Or a fox. Think of a fox. Wouldn't a red fox be really inconspicuous on a green background, except during the fall? Well, it turns out that the tiger don't see their own orangeness in the same way as the fox doesn't see it. From the fox point of view, it is brown, and the leaves are also always brown. From the fox's point of view, and more critically from the hare point of view, the fox and the leaves have exactly the same color. The mouse doesn't see a fox as having a different color than the background. And similarly, the plant hid their babies from mammals, and they were hiding in plain sight, while the birds could see them very well, because birds are tetrachromats and most mammals are dichromats, only have two prime colors. But then primates arrived, and primates lived in the trees, and primates eat lots of fruits. That's one of the main things that primates eat. Of course, if a chimp finds a squirrel or a bug or another monkey, sadly, it will eat it. So chimps eat everything, but most primates use fruits as a main food source. And because of that, it suddenly has become really beneficial for primates to reacquire this ability to see red on the green background. And there was this evolutionary pressure, and independently in New World primates and in Old World primates, trichromatism had started to reappear. But the process is really slow, and you can see, like, we are now living in these bodies for so long that we even stopped jumping into trees and learned how to grow our own produce, and still there was barely enough time to separate the sensitivity curves for green and red cones. This is interesting. And just as a final accord for this part of the lecture, let's look again at vision at birds and reptiles. I have already mentioned that they have 4 cones, so they're better than us. They have 4 cones. How many colors can they see with 4 cones? Is it just one extra color? What do you think? Well, let's figure that out. If you have 1 cone, how many colors can you have? Kind of 0, right? If you have 1 type of cones, then it's basically black and white. Everything is black and white. Okay, good. If you have 2 types of cones, like a sheep, you have short and you have medium. How many very different colors will you have? You have 2 primary colors, right? A person with 2 types of cones has 2 primary colors. For a person who cannot differentiate between red and green, blue and brown are still 2 different colors. So you have blue, you have brown, then you can have something where both of them are present. And when both of them are present, it's called the white. So the absence of color, which means that a person with 2 primary colors essentially has 2 colors and then some shades between them. A person with 3 colors, and that's us, we can have 3 primary colors. If we mix all of them, it's not a color, it's white. But now we can also mix them in pairs, right? So like blue and green gives you like teal. Blue and red gives the whole world of purples, and red and green gives you a new color, very new color, which is called yellow. Our landscape has 3 primary colors and maybe at the very least yellow and purple, but it means that a tetrachromat doesn't have just 1 color more. It has about 2 times more colors, because now so many combinations would become different colors. Think of that. If you have 4 cones, you have this color, color 1, color 2, color 3, color 4, but also 1 and 2 will be one type of color, but say 1 and 3 will be yet another type of color, and 1 and 4 will be yet another type of color. It's a little bit confusing, but think of that as a combinatorics problem, the problem of all possible combinations. It would be all individual colors, then all pairs, and all triads. All of them, in principle, could be perceived as unique colors. The point that I'm making is that there's a whole another universe. When a pigeon looks at the world, you literally cannot imagine how a pigeon perceives colors. It's not just one extra shade. It's like everything now either has ultraviolet added to it or does not. It's bizarre if you think of that. The world of a pigeon is so much richer than yours. But even now we only have started, because birds can also see light polarization. And I don't want to even tell you what light polarization is, because its physics is hard. But just believe me that light polarization is a thing. It's an additional property that light of any color can have or not have. If you heard of polarizing filters, they are used in photography to reduce reflections, because it turns out that reflections are polarized. So a bird, or many insects, as it turns out, can distinguish between colors that were reflected and colors that were not. Can you imagine that? And we don't know how they perceive them. Can they see objects based on whether they're polarized or not? Like if you have a polarized white object on a non-polarized white background, can you see it if you're a bird? I think the answer is yes, based on behavioral experiments, but that's a situation when imagining it and like stepping into the shoes of a bird becomes increasingly complicated. Arguably we cannot do that, but in this case I cannot even hint at some ways to imagine that. But even that is not all. To add to that, birds have a much better picture of the world overall, because they're also cursed by the same issue of all the supporting architecture approaching rods and cones from the wrong direction, but they solved this engineering problem in a different way. The evolution solved it in a different way. Instead of having a bunch of blood vessels coming through the blind spot and going like that everywhere, as it happens in us, birds have a special thing called a pecten. And I won't be able to give it justice, so if you like birds, go on Wikipedia. The link is again in the description. There is a whole chapter on bird vision, which is very well written and describes just how insanely cool birds are. If you think of a radiator in a room, I know this is a little bit of a jump, like in winter, sometimes there are those things in our rooms that make the air warm. Sometimes in the corner you would have a radiator filled with warm water, right? There would be a warm water tube coming to it and the tube come out of it. It's usually along the wall, often under the window. And this is the warm thing in your apartment that warms the air. Do you know what I'm talking about? Like a radiator. But another option is to have warm floors. To have a floor, and under the floor you would have tubes with warm water. Our eyes are essentially like warm floors, except all the tubes are on top of the floor and it makes it hard to walk. In birds, it's more like a radiator. They have a special structure in the eye that saturates the inside of the eye with oxygen and glucose. And because of that, they have no blood vessels on top of the retina. So they sacrifice vision in a bigger part of the eye. The blind spot is ginormous. It's not just the place where something enters the eye, but this entire thingy called the pecten. Here they don't have an image. But as a kind of payment for that, the vision in everywhere else is almost perfect, because they don't have any blood vessels running there. All of the blood vessels are sitting here and are saturating the inside of the eye with oxygen. And because of that, some birds can, for example, afford to have 2 fovea. Isn't it nice? If you're a hawk flying and then there is a mouse somewhere, I think the description again from Wikipedia is you can see an ant or a grain of rice from the 3rd floor, or like from sitting on the top of a tree. That's kind of the quality of vision in those birds of prey. They have 2 foveas, one to look straight ahead, and you also use it when you go down on a squirrel to kill it, and another one that you use to just leisurely see everything as you're flying in the skies. And on top of that, they also have colored oil drops inside their cones. So look, these are the cones that birds have, and they have special oil droplets over here. And that's not only true for birds, it's also true for crocodiles and turtles and tortoises. Here's how it looks like if you look at it under the microscope. Cones that otherwise would be similar to each other sometimes have different optic filters, these colored droplets of oil, potentially to even further enhance their color vision. At this point, we know that they exist there, but it's not quite clear what exactly they change about birds' vision. Still, it's super fancy. And this is one of the great examples when it's obvious that we have a clade, a whole branch of the evolution tree that is blessed with superior hardware. Everything about bird vision is better than mammalian vision. Well, maybe with one exception. One way in which humans are pretty good, we have a really balanced vision in the sense that we can see fairly sharp, but also fairly broad, and we can see in color, kind of, but also we can see at night. Many birds go blind at night because they don't have enough rods. They invest into cones too heavily. So our vision is overall pretty good as in the animal kingdom, but it would have been so much better if we had access to this machinery. On this note, let's probably call it, and see you in the next part of the Vision lecture series. Of pros and cons of rods and cones and spectral sensitivity.